Good job on micro exam number one. Really proud of some of those, your performances. Some of you gotta ramp it up, but for the most part, you did very well. Your mean was good. You didn't need any extra help to get your mean up to something. Stop. Nope, you did it on your own. Okay, so we went over this crazy overview slide last time, and we'll start to plow through the details. Development of your immune response. So this is starting to just move through the steps that the specific side of the immune response will go through as it gets to the place that it can mount a response, a defense against the specific organism that is now trying to do harm to you. Perceive and attach to non-self or foreign molecules. We kind of talked about this part of the immune system on the innate side. And on the innate side, what part of the immune response can help attach to non-self or foreign molecules? Complement, and which part of the complement cascade are we talking about? No. There's only three sides, so pick another one.
The other one's the inflammation, the inflammatory side, and the third side.
And the third side, that's the one we want.
Let's go, Bob. Come on, Bob.
Help me out, save us.
Come on, somebody help him out. He got 2 out of the 3. He tried that one first. Come on. Yeah! What did you say? What did we say? Opsonization, yes. Attaching itself to non-self organisms to make your phagocytic cells more efficient in going after it. And there's a specific number. What are the numbers that help that side of the complement system go? I like the 3 part. Not by itself. 3. No. You're good the first time. What'd you say the first time? 3A.
C3B? C3A, C4B.
Yes, the A factory, the 3A is what does most of the work of the opsonization. And since you brought up B, what's the B do in 3? C3B? Huh? Does C3B make more C3A and B? Come on, somebody. I know you haven't studied it, but hey, C3B along with C5 what?
Which one are we looking at?
We're looking at
C5A.
B, C3B
Okay.
along with
C5.
C5 what?
B.
Does what?
Does it interact with C6 and 7?
No. C3B and C5B goes into which arm of the complement system?
I'm looking.
Cytolysis.
Figure it out. Opsonization.
Huh?
Cytolysis.
All right, we need to do a little complement work. We'll come back to that. Go to another picture.
Slide 75.
There's C3B C5 splits C5A and B, C5B and C6 go down to this.
Oh, so it's optimization there.
What's this say?
C8.
It looks like they both go.
Oh,
It looks like they go
C3A
separate
goes over
places, doesn't it?
with C5A.
C3D does what?
Goes with C5, C3B is what's red. Oh, it goes over here.
And then what?
Optimization.
Which one?
Optimization, that's C3B.
C3B. Let's go, you guys have this one open?
Yes.
So let's get ourselves,
14.
chapter 14, slide,
75.
I don't have it open, so let's just get ourselves, reoriented on conflict. Is that the one you like? Everybody
Oh,
likes it?
75 is pretty, 74 is pretty good. That's just elected pathway though.
No, go back to that
Go
one.
back to the one I had.
Yeah, go back to what the slide number is that?
70.
All right.
70.
All right, I want you to, you can be our voice, okay? Go to, C3 convertase splits those things in two parts. What are the two parts that happens, then, after you split, you activate C3, what happens? C3A and C3B. All right, let's talk about the A side, since A comes before B. What happens with C3A? Okay, so C3A, I'll write it down. So we don't get confused anymore. Yes? Oh, please, fantastic. That's bothering you? Yeah. Hey. Let's see if I throw this here. I might have just written on myself too, but that's okay. Okay, so we got C3, and then we get C3B. Let's put it on, because I think that's how it is. Okay, C3A. C3B does what? Optimization, right? Along with what?
No, it breaks down C5.
C3B by itself, right?
Yeah.
On your little picture, it does more than that, but that's fine. C3 by itself goes to optimization. Okay, C3A, what happens there?
It combines with
C5A goes to information.
And what does it do?
information.
Cell thing, inflammation. So we're not confused anymore. We're back to our first bullet point on this one. I don't need these right. Okay, so major function of receptors. So perceive attached to non-self form molecules. We came out of the innate immune system that does some of these same kinds of functions in other ways. On the innate side, complement is what does much of this work. Okay. Promote recognition of self molecules. Why is it important to be able to recognize self molecules? So you know what not to destroy, right? And when that gets broken, exactly right, we have issues with autoimmune disease. When you lose the ability to recognize self molecules. To receive and transmit chemical messages among other cells in the system. We talked about chemical mediators, and we'll learn more about specific interleukins, which is a communication system between and white cells, and aid in cellular development. We talked about interleukins being able to cause expansions of clones of cells that are specific to the pathogen that is causing the invasion. So if one type of cell or if one cell line has been born to respond to a specific invader, that's trying to make you sick, it's going to be to your advantage for there to be a way to make more of those types of cells. And we'll show you that in more detail how that happens. Major histocompatibility complex. Okay, these are molecules that are found on all of our cells, all the cells of the body, all the nucleated cells of the body. So you see that red cells are excluded. There are no major histocompatibility complex molecules on red cells. Why? Because red cells don't have nuclei. That's right. So, in humans, the major histocompatibility complex is also called the human leukocyte antigens, or HLA antigens. And when do you hear about these HLA antigens, and when are these really, really, really important? Huh? Say it again? Tissue what? Yes, when you're transplanting. So if you need a kidney, and you're looking for a kidney that's most like yours, you want the kidney that's going to be transplanted into you to share many, 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 many of the same HLA antigens on its surface as yours. Because the more like your HLA system, the less your immune system is going to say, hey, this is foreign, we got to get rid of this thing. So that's what's going to determine your rate of rejection. Okay? Your own immune system is going to be happiest when you need a transplant, when the tissue that's transplanted is very similar to yours, and it's going to be looking at the HLA or major histocompatibility complex molecules on the surface. Okay, it plays a role in recognition of self by immune system and rejection of foreign tissue. That's just what we talked about. Okay, functions of the MHC when we're talking about immunity. Class 1, major histocompatibility molecules have the characteristic of regulating immune reactions mostly on the T cell side of the system. This is a generalization, but if you generally remember that MHC1 is associated with T cells and MHC class 2 are associated with B cells, you're going to be right most of the time. But notice that class 2 receptors are also found on macrophages and dendritic cells, and both macrophages and dendritic cells are APCs. What did we say APC stands for? Antigen presenting cells. So the antigen presenting cells and the B cells have class 2 major histocompatibility complex molecules on the surface. Okay. Now, this little picture there is of an antigen presenting cell. What has happened is, let's say this is a dendritic cell or a B cell. The B cell has determined that this antigen is the one that it was born to respond to. And we said B cells have what kind of receptors on their surface? What access receptors on B cells? They look like this.
antibodies.
Yes, they're antibodies. And so this one must have matched the antibody on the surface, and it was brought inside the cell. The antigen or the molecule or the bad guy is brought inside and is broken down into small little bits. The antigen is processed and it's presented on the outside of the cell. Okay, and it's presented on an MHC molecule. So if this is a B cell or a dendritic cell or a macrophage that's now presenting the antigen on the outside of the surface, it's what kind of MHC molecule are we talking about, 1 or 2? Huh? MHC class 1 or 2? 2. Yeah, you know, shout it out. So yes, if we're talking about an antigen presenting cell, we just said on the other side, these macrophages and dendritic cells are associated with MHC2. So the antigen is going to be stuck on an MHC2 class of a molecule. Okay, then we go again. Antigen presenting cells. Okay, you know I must have written this slide because it says stuff like chew up. Okay, it brings it in, chews it up, beats it up, processes the big antigen, breaks it down to small pieces, and it presents it to the outside of the cell on an MH2 class of a molecule. And once it's out there, T helper cells and B cells that are specific for that antigen can respond to the processed antigen. MHC class 1, again, on all nucleated cells, so we know it's not on what kind of cells? Right, it's not on any red cells. Okay, these present proteins produced by the cell, and these, they're all on the outside of our cells, so that our immune system can say, oh, that's an MHC1, we recognize that. That's a cell, keep moving, keep it moving. We don't want to mount any kind of immune response against that. So the immune system is normally monitoring all the cells in our body. They're looking at the MHC class 1 molecules to say, cell, keep it moving, cell, keep it moving, cell, keep it moving. But let's say one of our self cells has come into contact with a virus. We were sneezed on in an elevator. Some respiratory virus is now in the cells of our respiratory tract. So it brings the virus in, chews it up, breaks it up, and presents it on the outside of the cell on an MHC1. Now it looks different. The MHC1 is now changed because there's viral antigen being presented on the outside of the cell. That tells the immune system we don't have to be nice to this MHC1 class of cell anymore. We can destroy it because we know there's something wrong with it, there's something in there that we don't like. We can now destroy it because we see a different antigen on the surface of the MHC1. These cytotoxic T cells recognize there's something wrong with that MHC class 1 molecule now because it's showing viral antigen on the surface, and they can go and they can start destroying those cells to try to make us better. This sort of shows the difference between the type of molecules on a more molecular, cellular level. MHC1's on all human nucleated cells, MHC2 on macrophages, dendritic cells, and B cells. So peptides on the surface of our cells. Okay, lymphocytes. lymphocytes' role in surveillance. We talked about B cells having receptors on their surface. The B cell receptors are antibodies, and they will bind free antigens that are out there. They're only able to bind those with a specific, that fit the specific receptor on the surface of that particular B cells. All the other B cells will ignore them because it's specific, it's antibody, we know how specific antibodies are. T cell receptors process antigen with MHC molecules on cells that present antigens to them. Now we're going to see that the T cell receptors will respond to the antigen presenting cells, the antigens that are presented on the outside of our first responder, our antigen presenting cells, and the T helper cells will bind to those and then get the immune response going as they secrete interleukins out into the environment. Clonal selection theory, what in the world is this talking about? Well, the clonal selection theory is, I think of as like, you ever been to a casino, seen slot machines and, all those kinds of things? That's how I think of clonal selection theory. Lymphocytes have many, many, many genes. We have many genes in our host that are involved in making the specific receptors on the surface of our lymphocytes, B lymphocytes and T lymphocytes. 500 genes, that's a lot of genes to be in our slot machines. You pull the one-arm bandit and it spins, and whatever comes up is the receptor that's going to be on the surface of this B cell. You pull it again, and it turns, and we have a different possible receptor on the surface of the next B cell. So the possible combinations is just millions and millions and millions of different specific receptors that can be made from the different interplay of these 500 genes. Now, if you think about it, as you're in the casino, you pull the arm and it spins and spins and spins. There is going to be, just by statistics, just by chance, that you're going to come up with a receptor that matches a self cell. It just makes sense. It has to be a possibility that sometimes the combination is going to come up with a receptor that's going to recognize self antigens. In that situation, the body has a system in place to get rid of those cells, and so that they won't survive and be circulating in our bodies to potentially destroy the host. We'll talk about that a little bit, probably a few slides to come. Okay, so bone marrow, stem cells is where the stem cells are born. And we know that some will become T cells, some will become B cells, some will become monocytes and dendritic cells and all those kinds of things. The B cells are matured right there in the bone marrow. They don't have to go anywhere. The T cells migrate to the thymus. They're matured there. And then they move out into lymphoid tissue and they just hang out there. That's where they're going to be. Lots of B cells are going to be there. Lots of T cells are going to be there. Even when the thymus in childhood is going to regress, we still got plenty of cells that have migrated from the thymus and now are in the lymphoid tissues throughout our body. So seen this in another way. Bone marrow is where the stem cells are born. Some will become B cells, some will become T cells. B cells matured in the bone marrow, T cells in the thymus, and then they migrate to the lymph nodes. Okay. We talked about diversity already when we talked about the casino and the one-armed bandit and all those genes that we're working with to determine the specificity of the receptors on the surface of our immune cells. Now, since we have all of these millions and millions and millions and billions of possible receptors on the surface of our immune cells, that means we have that many possible antibodies, specific antibodies that we're going to be capable of making. The very cool thing about this system is that we have the ability, by the diversity of the receptors on our immune cells, we have the ability to respond to an antigen that has not even been invented yet. Now, I find that just absolutely fascinating. You hear about emerging infectious diseases, right? You hear about how viruses can combine their genetic material with this virus, and it can become this whole brand new thing that the human species has never seen. Or a bird virus has changed so that it can infect humans, or it can affect pigs, or whatever. We're not so worried about the pigs being affected by bird viruses, but humans, yeah, we're really concerned about that. But the immune system is ready. You know, their B cells are just hanging around and saying, "Oh, it's not my antigen again. No, it's somebody else's. Am I ever going to get a chance to play in the game? Am I ever going to get a chance to put on my jersey and get out there and make some antibody for my host?" Well, we can be encouraged to know that it doesn't matter what different kinds of mutations occur in the microbial world. Our body's ready. Our body, there is going to be a B cell there from all those 500 genes and all those pulling of the arm on our slot machine. We have it. There is going to be a B cell that's ready to get in the game for us. So that's the diversity of the immune system, and we're happy about that diversity because the microbial world is changing all the time. But we don't really have to be so concerned about our ability to make antibodies down the line. Memory, we talked about this being a good thing because once our immune system has been exposed to and has responded to and made antibody to a specific invader, infectious agent, the next time that we're exposed to it, we have this anamnestic response or a secondary response that's a very efficient response, which means it starts to put antibodies in the system very quickly. The B cell side of the line has memory, and the T cell side of the immune system has memory. So we have memory helper cells, we have memory B cells, and they just start to come to our defense very quickly. And many times it means we never even know that we were exposed to an infectious agent that made us so sick the first time that we were exposed to it. It doesn't mean that you never have any symptoms of the infection a second or subsequent time, but because of memory, you will be less sick. You may just, feel a little tired, you don't understand why you feel a little feverish, you just want a cup of tea for dinner instead of your, a regular, meal, but memory, is, a very wonderful, feature of our immune response. Okay? Again, same thing we talked about, just look at it in different ways. Our B cell diversity means that we have the potential in our bodies to generate more than 10 to the 10th different antibodies. Antibodies with different specificities, because we have that many different B cell lines with specific receptors on their surface. Really cool. Okay? When does all of this diversity happen? It happens in the embryo. The casino opens up long before the birth of the baby. These B cells are being cranked out with all these different specificities during embryonic development. I've used the term clone before, and it just means a B cell or a T cell that was born or programmed to respond to a specific antigen. Just one antigen or one epitope, okay? And when that cell, B cell or T cell, gets into the game, it creates more B cells that are capable of expressing that same specificity. So more players are on the team wearing that same jersey and same number. So that's what we're talking about when we talk about a clone, a lymphocytic clone. Third bullet point, different way of saying what we've already said. The first introduction of each type of antigen into the immune system selects a genetically distinct lymphocyte. It sounds very complicated, okay? But it's not so complicated. It just means the first introduction of an invader our body has never seen before is going to select out a lymphocyte that has that specific receptor on its surface and none other. All of the billions of different receptors on different B cells are very nice, but only one clone or one B cell or one T cell with that specific receptor on the surface is going to get into the game. So it's going to select out a genetically distinct lymphocyte. And when it does, the immune response expands that particular clone to be able to really nail that antigen. Yes, that looks very, 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 very crazy. Okay, but it's not so crazy. You'll see in a second. Okay, stem cell, bone marrow, embryonic life. That lymphocyte is inside of the casino and inside the slot machine. Okay, pull it, and all of these different combinations of receptors are going to result. Okay, with every pull, you're going to get a different receptor on the surface of the cells. Sometimes the receptor is going to be a receptor that will recognize self. It's just going to happen. Okay, and those self receptors are going to be eliminated. They're going to be eliminated by apoptosis. Those will not be allowed to remain. One of these must talk about that somewhere. Two at the same time out loud. number 2 says, lymphocyte clones that develop a specificity for self and that can destroy self cells in that host are going to be eliminated from the pool of diversity that we're making in the casino. And the process of eliminating those is called immune tolerance. We're going to tolerate our own antigen. Okay, so now we're happy with what has come out of the the casino. These are the ones that we're going to put into action. The ones that can recognize self are going to be destroyed by immune tolerance. Now, ready to go to work. We're born now, and now we're going to start being exposed to all kinds of antigens on our skins, on our caretakers, on our bottles, on breasts. However, you know, everything is going to have antigens that are going to now be introduced into the host for the first time. Now, enter antigen. The antigen will only be responded to by the clone that has that specific receptor on its surface. The rest of them are just going to pass by and say, oh yeah, nice to see you. I might get to play one day, but right now I'm not in the game. Once this one has bound to the specific line, we create a new clone, a whole clone of lymphocytes that are capable of responding to that antigen to make our system much, much more efficient. Better than having having five defenders is much better than having just one defender. And that's a clonal expansion. Now we're going to talk a little bit about antibody anatomy. They just look like Y's, you know, kind of these things that look like this. That's an antibody. That's what an antibody looks like. They're made of large glycoproteins. And remember, that first bullet point is reminding us that antibodies are the receptors on the surface of B cells. Just saying the same thing over and over and over. What is the receptor on a B cell? It's a immunoglobulin or an antibody. Okay, all antibodies are composed of 4 polypeptide chains, 2 heavy chains and 2 light chains. The heavy chains are the big ones, and the light chains are the little ones. So every once in a while, you know, logic comes and it helps us to remember things. So 2 heavy chains, 2 light chains to every antibody molecule, or immunoglobulin molecule. We say that those terms interchangeably. They look like a Y, and the end of the forks, like hands, that's the specific end. That's the end that binds to the antigen. On every antibody, there are variable regions and constant regions. Okay? The variable regions are at the tips of the Y's, like hands. These big ones, the big pieces are heavy chains, the little pieces are light chains. At the end of the Y's, at the arms of the Y's is the variable region, and that's where the specificity for antigen lies. So that's the region that's actually going to be binding antibody. So if this floor is the antigen, and I'm the antibody, in order to bind and defend against this antigen, I have to do a handstand. I can't stand up like this on it because it doesn't work. I won't be able to attach to it. The variable end that matches the receptor on the surface of the invader right here is down there. So That's how you bind your antigen with those ends. Another picture of what it looks like. Heavy chains are the big ones. Light chains are the small ones. The antibody binding sites are at the ends of the Ys, like hands down there. And there are disulfide bonds that cause the, they're not really Ys, they're all kinds of folds there. And there are disulfide bonds that hold the heavy chains together, that hold the light to the heavy chains as well. Now, notice that it says that this area, the FAB area, the antigen binding, that's what AB stands for, is up at the hands. And the feet, the FC, or the constant region, are the feet. And that area is what allows antibodies to cross the placenta. The feet is what binds to B cells. The feet is what binds to macrophages. So they stand up, like if I'm a B cell or if I'm a macrophage, my antibody on my surface are going to be standing up on their feet. They're going to be holding out their hands like this, trying to grab specific antigens out here. Okay? Wouldn't do us any good. It wouldn't be able to find things. It wouldn't be able to serve as a receptor if it was standing up like this. So the FC portion is what stands on macrophages or stands on B cells. We said B cells have the receptors are antibodies. They stand up like this, okay? So they can grab hold to the things out there that they have antibody to. Complement, remember it says complement binding site. We said that complement can bind antibody, and we'll talk about that more in a little bit. Same thing again. Yes.
I had a question. On
Sure.
that other slide, it said, well, I guess in the constant region, the ability to cross the placenta. I thought it was only a certain type of, I guess, antibody, immunoglobulin, IgG, that can cross the placenta.
Right.
So if you're saying that's constant, like.
Okay. Oh yeah. Okay. Let me tell you about constant regions. I'm probably going to say this again. I might have a slide for this. In fact, let's just wait. It'll make more sense later. Don't forget your question.
All right, no problem.
Make sure to keep me honest. Okay. This talks about diversity again, and it speaks to the difference between variable regions and constant regions. Again, more. I will not. This is interesting, and some of you want to know things in more detail. I will not test you in this much detail on the anatomy of antibodies. So you don't have to learn it in that much detail. Okay. Stop there. Okay. Just sort of gives you a look again at this thing that we call a Y. When you look at it more closely, you see it doesn't exactly look like a Y, but same thing. The constant region, the FC portion, is what binds to surfaces of cell, so that the variable regions are out and available to bind to antigens. Okay, we kind of talked kind of talked about some of this already. B cell maturation, we know where it happens. We know it happens in the bone marrow, and it just talks about what specific cells, the stromal cells of the bone marrow. They mature there, and once they have matured, they're sent out into other lymphoid tissues, the lymph nodes, spleens, gall, malt, salt, all those places that we've already talked about. So they put themselves in the way of the reticuloendothelial system, and as those fluids pass through, they'll bring different antigens with them. If someone has stepped on a piece of dirty glass, we like rusty nail. If we step on a rusty nail, that antigen is going to enter the reticuloendothelial system in our foot. It's going to come, remember, our reticuloendothelial system moves towards the heart, and it's going to pass through a lot of lymph nodes and stuff on the way. So they will come in contact with the specific B cells and T cells that are stored in the strategic places in our lymph nodes. So you can wake up and they can find the specific B cell that was born to react and defend against that particular antigen. Bless you. And you know, again, it's an immunoglobulin. It sits on those cells and with their feet, or the FC portion. Talked about this already, or at least some of it. Bone marrow is where their stem cells are. They go to the thymus. T cells are matured in the thymus, and then they also go out into the lymphoid organs. And there are CD4 and CD8 types of T cells, and we'll talk about the difference between the two. But that's what CD stands for, CD4 and CD8 clusters of differentiation. Some will become CD4, some will become CD8s, and then they'll be hanging out